For the last lecture in Unit 1, we're going to focus on the last standard to cover for Unit 1, which is nucleic acids. Our objectives in this lecture are to look at the function and purpose of nucleic acids. We'll look at their structure and how the double helix structure of DNA was discovered. We'll take a look more specifically at DNA structure and compare the structure of DNA to RNA. So nucleic acids are responsible for the storage, expression, and transmission of genetic information. So we talked about carbohydrates being energy molecules and structure in plants. We looked at lipids being long-term storage of energy, as well as being important in steroids and cholesterol. We also then talked about proteins being the main structural component of living organisms. Now, nucleic acids, such as DNA and RNA, are responsible for genetic information. There are two classes of nucleic acids. Deoxyribonucleic acid, which we commonly refer to as DNA, stores the genetic information encoded for the sequence of making proteins. And we'll learn about this, that DNA basically is a sequence of nucleotide monomers that has the code to make proteins. Ribonucleic acid, or RNA, is responsible for decoding the message in DNA and using that information to build a specific type of protein or polypeptide chain. More on that later, but I just wanted you to understand that these nucleic acids are in their own little lecture for a reason, because they do some remarkable things. And as you can tell, their structure is far more complex than any of the other biological macromolecules. Let's focus specifically now on nucleic acid structure. So there are some important structural features to nucleic acids. So the monomers that build nucleic acids are known as nucleotides. Now you may realize that nucleotides and nucleic acids kind of have the same prefix or word stem, and that is because they are found in the nucleus of cells. That is a theme we'll come back and touch on later on and really throughout this whole course. Now, a strand is a polymer of nucleotides. So remember, a polymer is a bunch of monomers together. So the nucleotides represent the monomer. The polymer is the DNA or RNA strand. Specifically in DNA, we have two strands that form a double helix, where it sort of resembles like a spiral staircase. More on this specific structure in just a bit. We also see that DNA in particular is wrapped around histones, which are proteins that act as spools that wraps DNA, like the thread of DNA, into nucleosomes. This is extremely important because DNA is a very long polymer. In fact, in every cell of your body, you have approximately 6 feet of DNA that needs to fit inside a microscopic cell and even a microscopic component of your cell. So that 6 feet of polymer has to be organized, and it's usually wrapped around, wrapped into nucleosomes and chromosomes. So let's take a look in the next slide. We're going to take a look at a video that shows how amazing this ability of DNA to condense around histones and form chromosomes in your nucleus, how important it is and how quite honestly amazing it is. So this particular video is called a molecular visualization. And so they've taken everything they know as far as DNA structure and behavior and made a computer simulation of what it may look like inside the cell. So it's pretty neat. So let's take a look at how DNA is condensed into nucleus.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell. The process starts when DNA is wrapped around special protein molecules called histones. The combined loop of DNA and protein is called a nucleosome. Next, the nucleosomes are packaged into a thread. The end result is a fiber known as chromatin. This fiber is then looped and coiled yet again. Leading finally to the familiar shapes known as chromosomes, which can be seen in the nucleus of dividing cells. Chromosomes are not always present. They form around the time cells divide when the 2 copies of the cell's DNA need to be separated.
More on cell replication and DNA replication later on in the course, but right now we're focusing on the structure of nucleic acids. So as we've mentioned, the monomer or building block of nucleic acids is the nucleotide. And there are some basic structural components of the nucleotides themselves. So these monomers are comprised of 3 main parts. First will be a 5-carbon sugar. Now this sugar could be deoxyribose, as is pictured here, or ribose. And this is where you see one of the differences between DNA and RNA. So DNA has deoxyribose sugar, so it's deoxyribonucleic acid, whereas RNA has a ribose sugar, naming it ribonucleic acid. Now, each nucleotide also has a phosphate functional group that you see highlighted there in yellow, as well as a nitrogenous base, which is highlighted in brown. Now, the nitrogenous base is very significant because there are actually 4 different types of nucleotides because of the 4 different bases they may possess. 2 types of nitrogen bases are known as purines. These are nitrogenous bases that have 2 rings. Adenine and guanine are the names of the 2 purine-based nucleotides. The other 2 nucleotides are known as pyrimidines, and these only have a single ring structure. Thymine and cytosine are the names of the pyrimidines. There are 4 nitrogenous bases that make up the structure of DNA: adenine, thymine, guanine, and cytosine. And we often abbreviate these bases with the letters A, T, C, and G. Now let's look at how we use those monomers, those nucleotides, to build a strand of nucleic acid. So nucleic acids have a linear sequence of nucleotides that have a definition. Basically, one end is called the 3' hydroxyl end, and you can see that highlighted down here. So the hydroxyl end, and you'll notice we say it's a hydroxyl because look, there is a hydroxyl group, an OH group, right here. The 5' end is called the phosphate end, and notice here a phosphate group is exposed at the end. So any strand of nucleic acid, whether it be DNA or RNA, will run in a particular direction. 5' to 3' is the adage, so 5' to 3'. We'll come back and touch on that later. Now, the strand itself is held together by what we call a phosphodiester linkage. This is a double linkage where you have 2 phosphodiester bonds that are holding together adjacent nucleotide molecules. So here is the phosphodiester linkage between 2 nucleotides. So notice the phosphate group will connect in with the exposed hydroxyl group, and that bond is formed because of a dehydration synthesis. And the same is repeated for this. So this basically builds the strand of a nucleotide, and we say that it runs from 5' to 3'. The backbone is basically the strand that includes the phosphates and the ribose or deoxyribose sugars. Notice the nitrogenous bases, the nitrogenous bases are extending out. They're exposed like spokes on a fork or as an open end of a zipper. This is significant. We'll come back and touch on this later. Let's talk about the discovery of the DNA double helix. This is something that was very significant as the race to the moon, because understanding DNA as the molecule for genetic inheritance was very, very important, not just for biology, but for the entire scientific community. One of the paramount discoveries and research points was done by this gentleman here, Chargaff. He did an interesting experiment where he analyzed the composition of the DNA, specifically the nitrogenous base composition in various species. And he found some very interesting patterns. No matter what species he looked at, he noticed that the percentage of adenine always seemed to equal the percentage of thymine. He found this in Homo sapiens, which is the Latin name for humans. He found this in the fruit fly, Drosophila melanogaster. He even found it in E. coli, a bacterial cell. Notice then also the percentage of guanine always tended to equal the percentage of cytosine. This led him to theorize that the adenine and thymine must be linked together in the double-stranded matrix of a DNA molecule. In other words, adenine must always be paired with thymine, and guanine must always be paired with cytosine. So if we look at this basic structure here we can see we have one strand of the sugar phosphate backbone in DNA, running from the phosphate 5' end to the 3' hydroxyl end. And notice the bases over here are adenine, guanine, thymine, and cytosine. The other strand of DNA The other sugar phosphate backbone is running in the opposite direction. More on that later, but notice the phosphate 5' end now is at the bottom and the hydroxyl 3' end is at the top. The bases that extend out and form with the other bases on the adjacent strand are complementary. If adenine is over here, thymine must be over here. If guanine is here, cytosine must be here. If thymine is here, then adenine must be on this side. And you'll notice that the bonds that connect these base pairs are hydrogen bonds. A little detail we'll touch on later is that there are always 2 hydrogen bonds in between adenine and thymine, whereas there's 3 hydrogen bonds between guanine and cytosine. Now, to remember the base pairs, there's a couple easy mnemonic devices. So A and T always go together. It spells the word at. I always remember that G and C pair together because the letters look very similar. In fact, G looks like a C just with a little extra line on it. So that always helped me to remember the base pairing rules that Chargaff discovered here. Now, the next scientist we're going to discuss is this woman here. This is Rosalind Franklin, and she worked with X-ray diffraction. So she worked with a lot of X-rays. And so what she did was she exposed DNA fibers to an X-ray and recorded some photographs. And so her image that you can see here showing that the 2 strands of DNA overlapped was very significant. Now, she didn't discover that DNA was a double helix, but she discovered that it overlapped in some way. And that information was very important for these 2 gentlemen, Watson and Crick, who were able to take Chargaff's rules, the base pairing rules, and Rosalind Franklin's X-ray diffraction photograph and see that DNA is organized into what's called a double helix. Let's learn more about how this was discovered by this group and team of scientists.
As late as 1951, the chemical structure of DNA remained a tantalizing enigma. At Cambridge University in England, biologist James Watson and physicist Francis Crick had been working to unlock the secrets of the DNA molecule. But they weren't alone. Several other groups of competing scientists were hard at work trying to solve the same puzzle. Some important facts about DNA were already known. For instance, scientists knew that DNA was composed of 4 bases: adenine, cytosine, guanine, and thymine. They also had been able to infer something about its structure with the help of X-ray crystallography. This technique involved passing an X-ray beam through a crystallized DNA molecule and capturing a vague, shadowy image of its internal structure on a photographic plate. Armed with this information, along with their own knowledge of chemical structure, Watson and Crick began building a 3-dimensional model of DNA.
I wanted an arrangement, where I had a big and a small molecule, and somehow you had to form link bonds. Here's A, and here's T, and I wanted this hydrogen to point directly at this nitrogen. So I had something like this. Oh. So then I went to the pair, and I wanted this nitrogen to point to this one. And I went like this. Whoa.
Today, you can buy a kit and assemble the structure that Watson and Crick put together. But we're getting ahead of ourselves. Watson and Crick needed more data. With the help of physicist Maurice Wilkins, they gained access to an X-ray picture of a DNA molecule that had been taken by his research partner. Her name was Rosalind Franklin. Without her knowledge, Watson and Crick used the data from that X-ray image and successfully completed their model. In a 1953 issue of Nature magazine, Watson and Crick revealed their amazing discovery to the world. Their model showed that the DNA molecule was a double helix. The twin strands were composed of pairs of the 4 known bases linked together by hydrogen bonds. And the whole structure corkscrewed like a spiral ladder, which could easily pull apart in order to make copies of itself with the same encoded genetic information. Watson and Crick had won the race. The discovery of the structure of DNA sparked a scientific revolution. It illuminated the molecular and biochemical foundation of life in a whole new way. It opened doors for areas of research and other great discoveries that few ever imagined possible. As for Watson and Crick, their discovery of the double helix, along with Maurice Wilkins, won them a share of the 1962 Nobel Prize. And what of Wilkins' colleague, Rosalind Franklin? Despite her contribution to the discovery, she wasn't considered for the '62 prize. The rules state that it can only be awarded to a living recipient. Rosalind Franklin died in 1958 of ovarian cancer, most likely caused by exposure to X-rays.
So the discovery of the DNA double helix was very significant. Now let's take a closer look at some of the details of the double helix. So it is an anti-parallel double helix. So that means that both strands run in a 5' to 3' orientation, but in opposite directions. And so we can see that here. Notice here, the 5' to 3' end is moving up the staircase or up the helix, whereas the complementary strand is going in a downward direction, down the helix. So they are what we call anti-parallel. You see that over here, 5' to 3', and then 5' to 3' going up. We also learned that adenine always binds with thymine, and it forms 2 hydrogen bonds. Cytosine then always pairs with guanine with 3 hydrogen bonds. So this is often why DNA, the DNA structure, is referred to in a couple of ways. It's referred to as a spiral staircase to reflect the double helix structure, the 3-dimensional structure of the molecule. But we also refer to it as a ladder, a DNA ladder, because the backbone kind of is like the sides of the ladder, and the hydrogen bonds between the bases are like the rungs. We also sometimes refer to DNA as a zipper because of its functionality. So what's interesting about DNA and these hydrogen bonds, if you remember, hydrogen bonds are weak but persistent. They're weaker than the phosphodiester and other covalent bonds that exist in the backbone. So because these hydrogen bonds are weaker, DNA, the 2 strands of DNA, can unzip like the zipper on your jacket and then reform, just like you can reattach your zipper together. That becomes very significant when we look at the function of DNA in the nucleus and also the function of RNA. Here's a little bit more on the structure of DNA.
The structure of the DNA molecule was first inferred by James Watson and Francis Crick, based primarily on X-ray crystallography data collected by Maurice Wilkins and Rosalind Franklin, and chemical analysis of base composition of DNA conducted by Erwin Chargaff. The key features of the structure are its right-handed double helical structure. Each helix consists of an alternating sugar-phosphate backbone with nitrogen bases projecting toward the interior of each helix. One complete 360-degree turn of the helix covers 10 bases of length and equals 3.4 nanometers in physical distance along the axis of the molecule. The width of the double helix is 2 nanometers. The nucleotide bases are attached inside each backbone of the molecule so that the nucleotides in one helix or strand are hydrogen bonded to the bases in the other helix or strand. The hydrogen bonds hold the two strands of the double helix together. An adenine in one strand is always paired with a thymine in the other strand, and a guanine in one strand is always paired with a cytosine in the other strand. Note that guanine-cytosine base pairs form 3 hydrogen bonds, while adenine-thymine base pairs form 2 hydrogen bonds. This makes guanine-cytosine base pairs more stable than adenine-thymine base pairs. Nucleotide pairing between strands also allows the sequence in one strand to determine the sequence in the complementary strand. The two ends of a strand are not identical. Note that on one end of each strand, a 3' hydroxyl group of the deoxyribose sugar is not involved in the backbone, or we say it's free, while at the other end of the same strand, the 5' hydroxyl group of the deoxyribose sugar at the end is free or may contain a phosphate that is free and not bonded to another deoxyribose sugar. This dissimilarity of the two ends of a strand creates the ability to uniquely distinguish each end of the strand. Because of this polarity of each strand, we can see that the two strands of DNA are oriented in opposite directions, or we say that they are antiparallel. In three-dimensional space, the two helices are not equally spaced along the axis of the molecule, such that a major groove and a minor groove are created by the asymmetric spacing of the antiparallel helix backbones. The major groove and minor groove play important roles in the binding of proteins that regulate gene transcription.
And finally, let's compare the differences between DNA and RNA. So as we've mentioned, DNA is named deoxyribonucleic acid, and DNA, of course, is the acronym to shorten that up. RNA is the acronym for ribonucleic acid. They have different names because of the sugars that are present. So DNA has deoxyribose sugar and RNA has ribose sugar. Another key difference is the presence of the base thymine in DNA. In RNA, thymine is not present and substituted with the base uracil. Now, we've gotten into the habit of using just the letters to signify these bases. So in DNA, we know we have A, T, C, and G, right? Well, in in RNA, we also have A, G, and C, but instead of T, we replace it with uracil. I always find it easy to just go to the next letter of the alphabet from T. So in DNA, you've got T. In RNA, instead of T, you just go to the next letter, U. U behaves very much like thymine in that it will only form a base pair with adenine. Another difference is in the number of strands. As we've seen with the DNA structure, it is a double helix. So there are two strands that are wrapped in this double helix shape. RNA is a single-stranded helix, as you can see pictured there. And one other difference is that DNA comes in one basic form. It basically is a strand, or sometimes a loop, as we see in bacteria, or ring of a double helix. But RNA, as we'll learn in a later unit, can come in different forms. In fact, there are 3 main forms that we'll learn about: ribosomal RNA, transfer RNA, and messenger RNA.